as Alex said, I study how organisms interact with their physical environment, and the particular interaction with the environment that I want to talk about today is smelling. And smelling is an ecologically very important activity. A lot of animals use odors to locate really important things in the environment like foods, mates, competitors, predators, and suitable habitats. And odors really are nothing more than molecules that are carried around in the fluid, the air or the water in the environment around an animal. So the first step in the process of smelling before the nervous system ever deals with any information about odors is the actual physical capture of those odor molecules from the fluid surrounding the animal. And we've been, for a number of years now, studying the physical process of odor capture from the surrounding fluid. One critical aspect of that process is how the nose itself interacts with the water or the air around an organism. But if we want to understand what the nose is capturing when it's doing its smelling, we also need to understand how odors are distributed in the environment, because fluids in the environment, water or air, move around. So what's the odor distribution like in the habitat where an organism is smelling things? And what I want to do today is put those two aspects together to address the following question, which is, how do the fluid dynamics of odor capture by a nose affect the signal that the animal intercepts about odors in its environment? So that's what I want to focus on today. And I'm not going to use a human nose. Instead, I want to focus on the hairy noses of crustaceans like lobsters. And the noses on lobsters are their antennules, and I'll get back to that in a minute. But before I go any further, I should share the blame for this work with a number of my collaborators. And I'm particularly happy to talk about this work at Davis because an inspiration for part of it came from some mathematical modeling that Angela Cheer did. She's a mathematics professor here now, but she was a postdoc in my lab back in the Precambrian, and did a model of fluid flow around cylinders that gave us a bunch of insights that have helped us understand how noses work. And I should also mention I did a bunch of experiments with cylinders and flow with two other postdocs, Kate Loudon and Barb Best. A bunch of undergraduates were involved in gathering data about various organisms. And I should also point out that Mark Stacy, who is a professor in engineering at Berkeley, did some mathematical modeling with us of mantis shrimp using data that a postdoc, Christina Mead, gathered. And the flume work that I'll talk about was done at the Environmental Fluid Mechanics Lab at Stanford in collaboration with Jeff Kossoff and some of his graduate students. So this is a very interdisciplinary project involving mathematics, engineering, and biology together, and a lot of people contributed. So let's get back to the organisms. And if you go to the fish market and look at the malacostracan crustaceans there, and malacostracans are just the tasty crustaceans like lobsters and shrimp and crabs. And if you watch them in the fish tanks at the market, what you see is they have a lot of busy little appendages at the front of their body that are wiggling around. And those appendages are their olfactory antennules, and they're flicking them through the water. And if we have a closer look at those antennules, what you see is that they bear chemosensory hairs called asthenotascs. And the hairdos on different noses are different from one species to another. So for mantis shrimp, we have a very simple arrangement with just 3 chemosensory hairs on each segment of the antennule. If we look at a crab nose, it's very different. It's a very dense array of hairs, much more like a toothbrush. And if we look at the spiny lobster, we find a very fancy array of chemosensory asthenotascs in this zigzaggy row, and they're in a cage of non-sensory guard hairs. So the arrangement of these chemosensory hairs is very different from one animal to the next. If we take a cross-section through one chemosensory hair and look inside, what you find is that they're chock-a-block full of the dendrites from chemosensory neurons. And so if we're going to get the nervous system involved in smelling odors, the first thing that has to happen is odor molecules have to get to these chemosensory hairs. So how does water flow through these arrays of chemosensory hairs when the antennules are flicking around the way the animals move them? And to understand this, I need to first go through some background because I figure I've got a diverse audience here and not everybody does fluid mechanics. So I want to ask you to do a thought experiment, which is to imagine you've got a big vat full of water, or you're in the bathtub, and you stir that water around and you watch what happens. And what you see is that the flow is very turbulent and messy and everything swirls around. And if you stop flailing and just watch what happens, that fluid has a lot of inertia and it keeps swirling for quite some time before that motion is finally damped out. So in that first thought experiment, there's water in the bathtub or in the pot. Now do another thought experiment that's a little kinkier. Imagine that you filled your pot full of honey, or you're in a bathtub full of honey, something very viscous. And what I mean by viscous is something that offers a lot of resistance to being sheared. And if you stir the honey up in the bathtub or in the pot, what you see is the flow is not turbulent. It's very smooth and laminar and orderly. And if you stop stirring, immediately the viscosity of that honey damps out any fluid motion and it stops moving right away. So in the first situation with the water in the bathtub, that was a flow situation dominated by inertia. And in the second situation with the honey, we have a flow situation dominated by the viscosity or the internal stickiness of the honey. And so whenever we think about a flow problem, the first thing we need to think about is how important is inertia and how important is viscosity in determining how the fluid moves. And there's a nice simple expression, the Reynolds number, that's the ratio of how big inertial forces are and how big viscous forces are for any flow situation. And it turns out it's a very simple expression. It just depends on some linear dimension. In our case, it'll be the diameter of one of those sensory hairs. It depends on how quickly the fluid is moving past the structure, like the hair. And it depends on the density and the viscosity of the fluid. So let's think about what this tells us biologically. If we're dealing with a large organism or structure like us or a whale or an eagle, and that structure is moving at a rapid velocity, then we're dealing with a high Reynolds number flow situation where inertial forces predominate, the flow is messy and turbulent, and it's the bathtub full of water situation we imagine. In contrast, if we think about a very small structure like a microscopic chemosensory hair, or something moving quite slowly, then we're dealing with a low Reynolds number viscous flow situation where the flow is very laminar and smooth. What's nice about low Reynolds number situations is that everything is very orderly and much easier to analyze. What's bad about low Reynolds number flow situations is that because you and I are big, high Reynolds number organisms, our intuitions are really lousy when we try to understand these small, viscous sorts of flow situations. So that's one piece of fluid dynamics you need to keep in mind. One other is something called the no-slip condition. And what that means is if we have a solid surface like the surface of a hair, and there's some fluid moving past it, and this arrow is just the velocity vector of the flow, so the longer it is, the faster the flow. Turns out that the layer of fluid in contact with the surface doesn't slip relative to the surface. It just sits there, and what that means is that there's a velocity gradient that develops between this stationary surface and the moving fluid. And it turns out the same thing is true if you've got a stationary fluid and you move a structure through it. The layer of fluid in contact with that structure moves along with it, and you get a velocity gradient between that moving body and the stationary fluid. And without getting into all the boring details of it, let me just tell you that the thickness of this layer of fluid that's kind of stuck to the object is greater relative to the size of the object if the Reynolds number is low. So that's another thing to keep in mind. The lower the Reynolds number, the thicker this layer of fluid moving with the object is going to be relative to the size of the object. So with this all in mind, let's go back to thinking about our arrays of hairs that are of finite width. And we can ask, does the flow go through the hairs like water would go between the tines of a pitchfork, so that we have a leaky structure full of holes, or is the boundary layer around each tine of this fork so thick that the fluid doesn't go through the gaps, it goes around the structure instead, so that we have a non-leaky structure. So if we want to think about flow around these chemosensory hairs, we have to figure out whether the flow is going to move between the hairs or not. And as you saw, a lot of those olfactory antennae had very different hair arrangements. So we want to understand which features of a row of hairs affect leakiness. And then this gentleman is here to help me remember to tell you the various features that we explored. Hairs can be narrow in diameter or large in diameter. They can be closely spaced or far apart. They can be short, they can be long. You can have a lot of them in a row or only a few, and they can interact with the solid surface nearby the animal's body. So we explored all those different factors in considering flow around arrays of hairs. And it turns out that this is a much more general and important question than just understanding hairy noses. It turns out hairy appendages are everywhere if you look across the animal kingdom. So we talked about hairy appendages capturing molecules, and here you see the feathery antennae that a male moth uses to sniff out where the girls are. Other very familiar feathery appendages that capture molecules, of course, are gills, like these feathery gills on a larval fish. Feathery appendages are also involved in capturing particles, and many aquatic animals, like this euphausid, capture single-celled algae and small particles using food particles using hairy appendages. And hairy appendages aren't just involved in capturing stuff, they're also involved in locomotion. So we find a lot of planktonic animals like this little larval worm swim around with hairy appendages. And even in air, tiny insects like thrips fly around with wings that have bristles but no membranes on them. So we find hairy appendages are everywhere in many different phyla, and they perform really important functions like gas exchange, smelling, feeding, and locomotion. And so for a functional morphologist like me, trying to understand how this basic category of structure, little hairs, operate will help explain an awful lot of biology. So how do these arrays of hairs work? And if you think about it, all the functions I talk about involve the interaction of the hair array with the fluid around it, so smelling, swimming, gas exchange. So how do these hairs interact with the water or the air? If you work out the Reynolds numbers of the individual hairs on these diverse structures, you find that the Reynolds numbers range between 10 to the minus 5 up to about 10. And what we know about that Reynolds number range is that we're dealing with laminar viscous flow. So imagine the bathtub full of honey for the rest of my talk as I explain how these structures work. And it's a little messier than just dealing with viscous flow, because in the Reynolds number range from about 1 to 10, we can't ignore inertial effects. So those come into play too. So it's a little more complicated than just a pure viscous flow situation, which of course makes it more interesting as well. So how do differences in structure and behavior of these appendages affect these functions? And the first step in addressing this was looking at flow around arrays of cylinders. And so I have to show you the model that Angela did as a postdoc. So this is an example of what you learn when you calculate what the fluid flow would be like between a pair of hairs if the fluid can go between the hairs or around the array of hairs. And what we've plotted here is leakiness, where a leakiness of 1 means that all the fluid that encounters a gap between two hairs will flow right on through that gap. So it's a very leaky structure. Or you can have a leakiness of 0, which means none of the fluid that encounters that gap goes through. All the fluid goes around the appendage. So the higher the leakiness, the more flow goes through the array of hairs. And we've plotted it here for hairs that are very close together and hairs that are very far apart. so that we can explore spacing. And each line you see is a different Reynolds number. And so the lower Reynolds numbers represent either very small hairs or very slowly moving hairs, and the higher Reynolds numbers represent bigger hairs or more rapidly moving hairs. And there's a lot of biology you can learn from a simple graph like this. For example, if we think about very small or very slowly moving low Reynolds number hairs, one of the things that we see is that changing Reynolds numbers, even by 1 or 2 orders of magnitude, makes almost no difference to leakiness. So these animals can move quickly or slowly, doesn't affect leakiness. They can grow by a factor of 10 or 100, and it doesn't affect leakiness, doesn't do squat. Also, changing hair spacing doesn't make any difference to leakiness. So at very small size, we have enormous permission for diversity of behavior, diversity of morphology, and it does not affect performance. Now, if we get to bigger and more rapidly moving sets of hairs, we find something very different. As Reynolds numbers get up to around 10 to the minus 1 or 1, we see that changing the speed that you move or growing has an enormous effect on leakiness. So just getting a little bigger or going a little faster can make something that wasn't leaky become very leaky. And the other thing that we see is that changing hair spacing in that Reynolds number range can also make an enormous difference to whether something is leaky or not leaky. So keep all that in mind, and let's think about what we learned about arrays of hairs. And so with Angela's mathematical models and our physical models, we explored the parameter space that this gentleman represents here. And I won't bore you with more graphs, but let me summarize what we learned. Hair diameter and spacing, as you saw in the graph I just showed you, only affects leakiness if the Reynolds number is greater than 10 to the minus 2. Makes no difference at smaller size. But if we look at a different morphological feature, that is how long the hairs are, we find something very different. We find that hair length only affects leakiness at Reynolds numbers lower than 10 to the minus 2. But it doesn't matter at the higher Reynolds number range. And then we find something very weird if you look at the number of hairs in a row. If you have a row of hairs and you put more hairs at the end of the row, if the Reynolds number is greater than 1, the structure becomes more leaky, more flow between the hairs. But if the Reynolds number is less than 1, the structure becomes less leaky if you put more hairs in the row. And so what we see is which morphological features affect performance depends on the size and the speed of the organism, that is, on its Reynolds number. And we also find that the consequence of a morphological change is different if an organism is a high Reynolds number or a lower Reynolds number. So this is weird. Most biologists are used to thinking about a particular structure having a particular performance consequence. And what we're discovering is that it really depends on the size of the organism. Now let's consider behavior instead. That is how the organism moves the appendage around. And as you saw in the graph I showed you, hair speed, that is Reynolds number, only affects leakiness if the Reynolds number is greater than 10 to the minus 2. But if we think about a different behavior, which is, do you move your appendage close to a body surface or far from a body surface, we get a very different result. We found that the motion of a hair array near a wall only affects leakiness if the Reynolds number is less than 10 to the minus 2. So different behaviors affect leakiness at different Reynolds numbers. And so what we see is that as Reynolds number changes, that is, as an organism grows or as it changes its speed, we get transitions in how these hairy structures work. Something that functions as a paddle that isn't leaky at low Reynolds number can become a sieve if it moves a little faster. We find that which structural features are important to performance changes and which behavioral features can affect performance changes. So this is all a little weird, but keep it in mind because it will help us understand noses. Okay, so let's apply these principles to flow through the arrays of hairs we find on hairy antennules. And let's start out considering the example of the olfactory antennules on spiny lobsters. So let me show you what a spiny lobster does with its antennules. So here's, you see there are two branches to the antennules, and the hairs are only on this branch. And you can see this is the only branch that's flicking. So the animal will move its antennule somewhere and then go flick, 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 and move its antennule and go flick, flick, flick. And it's very important that they keep the gunk off of their hairs, and you're about to see a lobster picking its nose. They spend a lot of time grooming those hairs to keep them clean. And so here he is. This is a lobster nose picking. So that's what lobsters do if you just watch them in the tank. But in order to figure out the details of the flow, we need to quantify that motion a little better. So we did high-speed videos of the antennules flicking. This is the hairy branch of the antennule. The lobster is sitting down here, and I'm going to show this to you slowed down about 8-fold from real speed. And so here it flicks, and then it returns to its original position. And so what I hoped you noticed is that the flick was very rapid, but the return was much slower. And if I can show you that again, watch this. Oops, it didn't work. Let's try it again. Nope, didn't work. But anyway, the flick was very rapid. The return stroke was much slower. And in fact, if you work out the Reynolds numbers of the individual chemosensory hairs, during the rapid downstroke, a hair has a Reynolds number of about 1, and during the return stroke, the Reynolds number is about 0.1, so much lower. And if you go back and look at Angela's calculations again, let me remind you, we're working in this Reynolds number range. This is the flick, this is the return. And this is a Reynolds number range where changing speed has an enormous effect on leakiness. So just from this general model, we would predict that that hair array ought to be leaky on the flick down and not leaky on the return. So odors ought to penetrate the hair array just during the downstroke is what we would predict from what we know in general about arrays of cylinders. We did similar measurements on a different species of lobster, the clawed lobster, and here are its olfactory antennules, and we found the same thing, that the downstroke of the flick happens at a Reynolds number of about 1. We'd expect the array of hairs to be leaky. The return stroke is slower at a Reynolds number of 10 to the minus 1, and we would expect the hair array not to be leaky. And if you look at lobsters as they grow, one of the things we found that was kind of cool is that they maintain the this Reynolds number relationship. Even little guys and great big guys all do their downstroke at a Reynolds number of 1 and a return stroke at a Reynolds number of 10 to the minus 1. So little lobsters have disproportionately big nose hairs and flick a little faster to maintain this Reynolds number as they grow. So we're predicting in general terms that the flow around the hairs should be leaky during the downstroke and not during the return stroke. But to really test this prediction, you have to get in there and measure the water flow between the hairs during the downstroke. And as you could probably tell from those videos, making measurements on microscopic hairs of things that are moving very rapidly is quite difficult. So to test this, we took advantage of a nice trick that engineers and biologists can use, and that is using dynamically scaled physical models. And what I mean by a dynamically scaled physical model is if you build a scale model of, for example, a lobster antennule, and you keep the Reynolds number of the model the same as the Reynolds number of the real antennule, then the ratio of inertia to viscosity will be the same for the model as for the real hair. And it turns out that the distribution of velocities around the model will be the same as around the hair, around the real antennule, and the force distributions will be the same as well. So this is very convenient for us. What it means, I'll remind you of Reynolds number, is we can make a large model of some dinky little microscopic structure, and we can move it at a nice, reasonable velocity that's easy to control and measure. And if we move it in some disgusting viscous fluid like Koehl syrup or mineral oil, then we can have a model that has the same Reynolds number as the real antennule, and we can study the flow around the model. So here are models of sections of the nose of a spiny lobster and a clawed lobster, and we towed these models through a big vat of Koehl syrup that I have in my lab at the same Reynolds number as the real antennules operate. And we put neutrally buoyant particles in the Koehl syrup so we could see how the fluid was moving. And when you do that, it looks like a snowstorm. And it's very confusing. So in order to actually measure how the fluid is moving around those hairs, what we do is shine a sheet of laser light through that vat of Koehl syrup, and what it does is make an optical slice through that whole tank so that you just see the particles in one plane at a time. And let me show you some examples of what we learned when we did that. So here's a model of a spiny lobster antennule, and the antennule itself is poking out into the room towards you, and the hairs, the chemosensory hairs, are poking out to the side here. Here are those guard hairs. So the antennule is flicking down, and that means that the motion of the fluid relative to the antennule is moving up. What you see here are the positions of individual marker particles on successive frames of the video. So these dotted lines show you where the particles are moving, and the further apart they are, the faster they're going. And what you can see is that fluid moves in the one side of the ask-the-task array and out the other side. There is indeed water motion through the array of ask-the-tasks during the rapid flick downwards. So they are leaky and odor-bearing water can get in. Here's the antennule on the upstroke, so it's moving in this direction. The water movement relative to it is this way, and what you can see is that the water does not flow through the ask-the-task array, it flows around it. So again, our prediction that the water would not flow through the hair array during the return stroke is true. We did similar experiments for mantis shrimp. Here are their antennules, and we found exactly the same thing with our dynamically scaled physical models, that when they do their flick, water moves into the array of ask-the-task. When they do the slower return stroke, the water that flowed in there remains trapped between the hairs. And we found the same thing for crabs, and here are the little noses on the crabs. And remember, crab noses look like a toothbrush with very densely packed chemosensory hairs. And the crabs do a second trick in addition to changing the velocity of the flick and the return stroke. And let me remind you again on Angela's graph, we're dealing in this Reynolds number range where the flick is at a Reynolds number approaching 1. I won't compete with that guy. Remember, in this Reynolds number range, if you change the spacing between hairs, it makes an enormous difference to how leaky the hair array is. And now let me show you a crab antennule. This is an end-on view of that toothbrush. Here are the chemosensory hairs, and the antennule is flicking this away. And when it flicks, you can see that the hairs splay apart and the gaps between them become bigger, so leakiness increases. During the return stroke, when the antennule moves back this way, you can see that the hairs, which are flexible, collapse back together again and are much closer spaced. So both the change in speed and the change in hair spacing contribute to the antennule being leaky here and not leaky here. So to summarize what we learned for various species of crabs, shrimps, and lobsters is that their flicking antennules operate in a Reynolds number range where changes in flicking speed and changes in hair spacing have an enormous effect on leakiness, and we would guess, therefore, in molecule capture, but I'll get to that soon. And so what we find is that flicking represents pulsatile sampling of the environment. Flicking is sniffing, so each time you see a lobster or a crab do this, they're sniffing. So they sniff here and sniff now and sniff later. So they're not continuously sampling their odor environment. They're taking sniffs. Okay, so that's the fluid mechanics of the nose, but what is the nose catching when it does a flick? And so to consider that, we need to look at how odors are distributed in the moving fluid in an animal's environment. So if you go out to, for example, a coral reef habitat where spiny lobsters might live, and release dye, which is another dissolved molecule in the water, and watch how it moves in the habitat, you get a good idea of how odors are dispersed. And when you do that, in this case, the dye is a fluorescent green dye. You see that as the currents move across the habitat, the odor is transported across the habitat. And as it goes, because the whole habitat is very large with rapidly moving flow, it's high Reynolds number, the flow is very turbulent and messy, and as all those eddies swirl around, the odors get dispersed and mixed into the surrounding fluid. So a plume of odor gets bigger and wider and more dilute as it moves across the environment. So that's how an odor plume looks to us. We are large organisms with big noses, but how does the odor distribution look to the nose of a crab or a lobster? What's the odor distribution like on the scale of one of those antennules, which we're dealing on a scale of millimeters to centimeters? So how are we going to have a look at odor distributions on that scale? And those of you who heard my talk yesterday heard about the technique that we use, but let me describe it for those of you who weren't there yesterday. So let's first think about the scale we want to sample the odor plume on. And this is a view of an antennule of a lobster, and the antennule itself is about 1 millimeter wide. Here are the chemosensory hairs and the guard hairs, and when the antennule flicks, it basically slices through the water column and only samples a little bread slice 1 millimeter thick out of that whole odor plume. It's not seeing the whole odor plume. So that's what we want to do. We want to take a little 1 millimeter slice out of an odor plume and look at it and look at the concentration distribution in that slice. And so how do we study that slice? Well, the first thing we have to do, we're going to study it in the lab, but we want our lab situation to mimic what water flow is like in the field. So we have to go out into the field with our flow measuring devices and measure water velocities where the lobsters actually live as a function of time. And so you get a time record of water velocity, and what we're really interested in is the turbulence, the swirling eddies in the flow, which is what's going to mix the odor up. And so we need to characterize that. And if you look at a plot of water velocity as a function of time, it's a very wiggly plot. The velocity is changing a lot as all these turbulent eddies sweep through. And we can characterize the turbulence by asking how much of the variation in turbulence happens at low frequencies and intermediate frequencies and high frequencies. And so by working out a spectrum of the variations in velocities at different frequencies, we can characterize the turbulence of the flow. And with that information, we can then go into a flow tank or flume in the laboratory and recreate similar turbulence there. So we did that. I went down to the Environmental Fluid Mechanics lab at Stanford with my data, and those guys set up a turbulent flow that mimicked what I had measured in the field. And here you see a lobster. I hope you can see with the light in this room. Here's a lobster in a flow tank, and it's about 1 meter downstream from an odor source, which is over there. And we can mix the odor with a fluorescent dye so we can see it, and the brighter the dye, the higher the concentration of the odor will be. And we do that, and here's the flow direction, lobsters facing upstream. Basically what we see is a big cloud, just like we saw in the field. And remember, what we want to see is just 1 millimeter slice through that cloud, which is what the antennule would encounter. And we do that by shining a sheet of laser light through this cloud of dye, just like we did in our tank of Koehl syrup to look at one slice at a time. So the antennule flicks through a millimeter slice of water, and we use the laser to look at only that slice. So here's our lobster again, and it's illuminated now with a sheet of laser light, a millimeter thick, comes down here, bounces off a mirror, and is reflected up that way. And when you shine a sheet of laser light through the odor plume, you see it's not just a diffuse cloud. You see that there are filaments of very high concentration. The brighter and lighter this is, the higher the concentration. And they're swirling around in water that isn't stinky and doesn't have odor. So it's not just a diffuse cloud. And here is the antennule flicking through this. So if we follow the antennule, we can measure the concentration it encounters as a function of time. And here's our very cooperative lobster that is flicking with its antennule right where we have our laser sheet shining. So how on earth do we get a lobster to do that? We did that by using a robotic lobster. So this is the shed exoskeleton of a lobster that we used to build our robot, and it has a computer-controlled motor which flicks its antennules. And what we would do is remove antennules from fresh lobsters we got at the fish market and put those real antennules on our robot, and then we could get it to do however many flicks we wanted, wherever we wanted in our flume. And so let me show you a video of what a lobster nose actually encounters. So what you're going to see here, here's the hairy part of the antennule of the lobster, and the brighter these stripes are, the higher the concentration, and the flow is going to be moving this way. So watch what happens when the lobster flicks, and remember it's only catching odors during the downstroke of the flick. So here, caught something, nothing, no smell, smell, no smell, got a good one there, nothing, smell. So not every sniff, even though he's in the middle of the odor plume, not every sniff encounters odors, and the pattern of stripes of these odors is different with each sniff that the animal takes. So that's what happens when a lobster is sniffing. So what we learned is that ambient flow around lobsters in the real world is turbulent, and in turbulent flow, on the scale of the antennules, the odor is in filaments of high concentration swirling around in unstinky water. And so one question we might want to ask is, does the spatial pattern of filaments in a turbulent odor plume depend on where the animal is located relative to the odor source? And that's been investigated by a number of people interested in turbulence, and let me just show you an example from some of John Cromaldi's work. This is a picture of what a turbulent odor plume looks like very close to the source, about 30 centimeters downstream from the odor source, and compare that with what the plume looks like further away from the source. And if you go in there and quantify this, what you find is that the filaments become wider, the concentration gradient at the edge of a filament is less steep, and the peak concentrations are lower as you get further away from a source. So there's a lot of information in an odor plume about where an animal is relative to where the source of the odor is. And so can animals use this information? And one step in using the information is, can they capture that information, or does flicking just stir it all up and make a mess? So the pattern of odor filaments contains information on distance from the source and also on whether you're in the center of a plume or on the edge of a plume. The filament structure changes. And so there's spatial information there. Can the antennules capture that information? And to test that, we made more high-speed videos of antennules on our mechanical lobster flicking in odor plumes, but we made high magnification videotapes just looking at the hairy part of the antennule, and we made high-speed tapes so we could really see what was going on. And here's an example of a frame from a video. Here's the antennule. Here are the chemosensory hairs. It's flicking in this direction, and in this slide, the ambient flow is moving this way. And the brighter these dye filaments, the higher the concentration. And what you can see in this particular video frame is that while the antennule is flicking and is leaky, these filaments of odor are moving up into the hair array. And what we did to follow what an antennule was capturing as a function of time is in each frame of the video, we would run a transect through the chemosensory hairs and measure pixel brightness, which was calibrated to odor concentration, along that transect. So we did that from one frame to the next, and let me show you what we learned when we did that. So this is a map of how the world smells to a lobster nose. This is position along the antennule. This is the hairy part of the antennule near the base and out near the tip, plotted as a function of time. And here a flick occurs. And what you can see at this position, all the bright colors indicate high concentration, so you can see a filament of high concentration, another one here. The darker areas show lower concentration, or black is no odor. And so you see that during the flick, the animal picked up a pattern of stripes and held on to that pattern of stripes until the next flick. During the next flick, the old water gets cleared out. It picks up a new pattern of stripes, which you can see here, and it hangs on to that new pattern during the return stroke when the water's trapped between the hairs, and while the antennule is paused when the water is trapped there. And it doesn't get a new pattern of odor till the next flick when the array of hairs is leaky. And you can see now it picks up a different pattern of odors, hangs on to it, and then flicks again, gets a different pattern, and then flicks again and gets a different pattern. So each sniff of the antennule picks up a different pattern of odor filaments. So how does concentration at a point, for example, concentration where a particular sensory, chemosensory hair is, vary as a function of time? And we can address that by just following what happens with time at a particular position. And let me show you the kind of thing we can learn when we do that. This is a plot of concentration as a function of time, and this is at a point, so you can think of it, what's arriving at a particular ask the task, during the stationary pause when no water is flowing through that chemosensory array, and you can see the concentration really doesn't change. In contrast, when the animal flicks and the hair array is very leaky, what you can see is that a filament came through, washed away, another one is coming through. So the concentration changes when the water can flow through the hair array. And then during the slow return stroke, this low concentration that was in the hair array right at the end of the stroke persists. So this is just further evidence that the only time that water and odors are moving through the hair array is during the rapid downstroke of the flick. So then we might ask, is this odor filament sampling pattern really due to the resistance of the hairs to the movement of fluid through them? And what role do the guard hairs play? And what role does the distribution of the asthetasts themselves play in determining this? So we can do some experiments to address this. So here's a scanning electron micrograph of the guard hairs and the chemosensory hairs on a section of an antennule. We can go in and snip off all the guard hairs so that all we have left are the asthetasts, and we can repeat those same experiments in the flume and see what happens. And here's what we found. Here's the graph I just showed you for an intact antennule. Here's an antennule that's had a haircut and has no more guard hairs. And what we see is that in both cases, during the flick, the concentration varies a lot as odor filaments wash through the hair array. But during the return stroke and the pause, when no fluid moves between the hairs, the concentration is steady. So there's no effect of those guard hairs on the sniffing pattern. And we can then ask, what about the asthetasts themselves? So we can completely shave all the hairs off of an antennule and repeat the experiment again. And here are the two graphs I just showed you with an intact antennule and asthetasts only. And let's compare that to a case where we have no hairs at all. So all we have is the stalk of the antennule flicking. And we can also compare that to a virtual antennule where there's no antennule at all, but you simply move a transect through the flow and see what it would encounter if it were moving the way the antennule was. And when you do that, what you find is that during the flick, during the return, and during the pause, the concentration varies a lot. In other words, there's a lot of flow past that structure all the time. And so this just corroborates what our earlier experiments told us, which is that flow and odors only move through the hair array during the flick, and it's the spacing of the asthenotasks, not the guard hairs, that is what's important. So if you remove those closely spaced hairs, the animals lose the ability to sniff. So let's sum up what we know so far. When an antennule flicks, water penetrates the hair array only during the downstroke, so the animal is sniffing. And the asthenotask array is able to capture a snapshot of that fine-scale filamentous structure of the odor plume when it takes a sniff, and it hangs on to that sample until the next sniff. So it's taking discrete samples of its environment in space and time. So it's now got this snapshot, but what are the odors getting to the asthenotasks? And so we have to now look within the asthenotask array at how the odor molecules are diffusing. So when an animal sniffs in a turbulent odor plume, what's the flux of molecules to the actual chemosensory hairs? And we examined this question using mantis shrimp because, remember, they have a very simple hair array on their antennule. And so they have only 3 chemosensory hairs on each segment of the antennule, and we used dynamically scaled physical models to work out the detailed velocity field around those hairs. So imagine a slice through 2 rows of chemosensory hairs, and the flick is in this direction. These guys flick with the hairs poking out to the sides, like so. So the flick is that way, the flow relative to the flick is that way, and we mapped the velocity vectors around all these hairs during the flick, during the return stroke. And then what we did is we took our flow data to Mark Stacy, a mathematical modeler, who then did what's called an advection diffusion model of what was going on there. And let me explain basically what he did. What you're seeing here is the antennule. These dotted lines show where the edge of the antennule is. The chemosensory hairs are poking out towards you. You see them in cross-section, and there are 3 rows of them. The antennule is flicking down, and so that means flow relative to the antennule is up in the direction of these yellow arrows. And what we did is then put an odor plume, a filament from an odor plume, in the water that the antennule is going to flick through. And the color scheme here indicates concentration. So this is based on our measurements. And then what Mark did is he let this odor filament get carried through this hair array in the flow field we had measured. And as it goes, the molecules are undergoing molecular diffusion. And so here's a little bit of time later, and you can see the molecules are diffusing, and the stripe is being deformed as it moves through the hair array, and here is later. And what he did was sit on individual asthenotask surfaces and ask, what's the flux of molecules to the surface as a function of time when the antennule is flicking? And here's the kind of result that his calculations showed. This is the flux of molecules to the surface of an individual chemosensory hair plotted as a function of time during the leaky outstroke of the antennule. And what you can see is that the flux increases very quickly, reaches a very high peak, and then tapers off very quickly. He can do the same sort of calculation for the return stroke. So when the antennule hair array is not very leaky, if the antennule were to encounter that odor filament, then you get a very different result. There's a very slow onset of concentration as the molecules diffuse in through that trapped water, and the peak is not very high at all. So when we're interested in the rate of molecule delivery to neurons, what we need to mimic is this high, rapid onset and high peak, because this is what's realistic for what real chemosensory hairs would be encountering on a living animal in the real world. So let me stop and sum up what we know so far. If we ask, what's the fluid flow like near a moving olfactory organ on one of these crustaceans, we see that flicking antennules alter their leakiness so that only during one stage of the flick do they sniff or take a sample of their environment. If we ask, what's the odor distribution like in the moving fluid environment of an animal, if we sample it on the scale that the animal's nose samples it, what we see is that in that turbulent flow out in nature, the odor distribution is filamentous, and it varies with distance from the source, so there's information in that plume. And if we ask, how does the physics of odor capture by the olfactory organ work, and what does that do to how the world looks to that olfactory organ, what we see is that the physical process of capturing the odors filters the odor information from the environment that the animal actually encounters. So that the animal doesn't see a continuous picture of the plume, it sees discrete snapshots of that plume. So the physics of catching an odor is the first step in processing odor information from the environment before the nervous system ever gets involved in further filtering of that information. Well, so what? Who cares how hairy little antennules work? And I would argue that there are a couple of so whats. One so what is that I think that people who study the neurobiology of olfaction need to remember that nose physics comes into play before the nervous system does, and that odor should be delivered to the neurons they're studying in ways that mimic the way that they really would be delivered to the real organism, and also that the physics of the nose is a big important step in filtering information from the environment. Another so what is that what we've learned about how noses work in turbulent marine environments has provided inspiration for the design of man-made noses, that is chemosensors, that they're designing to put on robots that they can send in to clean up toxic waste or mines or things that where you don't want to send a diver, but you do need to be able to find the source of the odor. So those are some so whats about noses, but for me, the real so what is the bigger picture, which is that hairy appendages are everywhere, and they perform important functions besides just smelling. They're involved in gas exchange, feeding, swimming, flying, and so the principles that we've learned by studying hairy noses also apply to a lot of these other structures and will help us understand how the different morphologies of these kinds of appendages affect their performance as well. And so I'd like to end just by saying that we biologists love the diversity of form of creatures that we study. And if we just use some simple physical principles as we look at this diversity, sometimes we can see general rules that will help us understand seemingly very different things, like the nose of a lobster and the wing of a thrips. So I'll stop there and see if you have any questions.
So it was a very nice demonstration of the spatial information present in the plumes. Have you or anyone else or your collaborators measured to see whether these animals can actually behaviorally determine the distance?
Yeah, so I actually, let me just tell you what we're doing. We've been, Christina Mead and Meg Wiley have been working with stomatopods which are small animals so that they can move around a lot relative to the size of a flume, which might be about 1 meter wide. And what we've been doing, they also flick laterally rather than vertically. So we can shine a sheet of laser light laterally through an odor plume. Here's a mantis shrimp. Here's its antennule. And we can record exactly what each nose is capturing every time it flicks, as well as record the behavior of the animal. And so this work, we're in the analysis stage right now. And so what we're trying to do is figure out what the search algorithms are that the animals are using, because now we actually know what each nose is catching each time it sniffs. So we're trying to correlate the behavior they do next with what each nose just caught. So I don't have the answer yet, but we're working on it. Earlier studies where people have studied things foraging in plumes, they've just looked at the whole plume. So they think an animal is in a plume or out of a plume. But here an animal is in the middle of the plume, and you can see that this nose isn't smelling anything, even though it's in the plume. So we're working on that. And then the other thing we're doing is we're working with Barry Aki, who's a neurobiologist who works on spiny lobster. And he's got a postdoc who's recording out of the olfactory lobe while we're recording what antennules are encountering as they sniff. So we had to make a special flume that had no motors so we could create our turbulent plume. We're also trying to see if the brain does anything different when the antennule encounters different patterns of odors. So that's a long answer to your question, which is we don't know yet, but we're trying. Oh, yeah, Lou had. Oh, okay.
So I guess along those lines, I was sort of thinking about it from maybe the other direction. Can you construct experiments where you actually fool them? That is, they exhibit a behavior that they would normally exhibit. I was thinking about changing the turbulence, that if you put them in a different turbulence environment, release an odor, do they act differently?
We haven't done that yet, and that would be really interesting to do because we can see how the structure of the plume would change.
Exactly.
Yeah, so we haven't done that yet, but that would be a lot of fun to do. One thing we have done, however, is we've done the stomatopod experiments in unidirectional flow and in wavy flow. And in wavy flow, the flow is reversing direction, so upstream is not always the same direction in waves. Yet these animals are able to find odor sources in their natural habitats, which are shallow and exposed to waves. And the turbulent structure is a little different in waves. And so we're doing the same analysis that I mentioned before, where we're looking at exactly what the antennules encounter and what the animal does next. And it's looking in our initial analysis like they're following the same rules in both situations. And what happens if they're going the wrong direction in the waves, then as they move further away from the odor source, what the antennules encounter is a little different. And if they follow the local rules, they get themselves turned around and go back in the right direction. So they do correct, they'll go wandering off in the wrong direction for a little while, but once they get far enough away, they figure it out and turn around and come back. So I hope I'll have more details about that soon. But it'd be fun to just stir the water up more. Make the same velocity of flow be more turbulent and see what happens.
It seems like you could change the turbulence by changing the viscosity slightly, maybe. So
you can do that. The other thing you can do is there are ways you can trip the flow upstream in the flume, put little things in the way that will shed eddies and change the turbulence. So, yeah. Other questions? Yeah?
So Barry's group has shown the refractory period of the sensory neurons is around 100 milliseconds, and has timed that to the flick rate. And I was wondering why you use such a slow flick rate and not, or not a, how do you say it, it's a stepped flick, or maybe you can try a step.
Well, the flicking rate that we used were the average downstroke duration, return duration, and the average pause between flicks. So what our spiny lobsters do is they'll go flick, 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 flick in one place, and then they'll move somewhere and go flick, 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 flick. So what we mimicked was the flick, 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 flick in a row. And so the timing that we used was what we measured on the real animals. And what I think is going on is they're hanging onto the odor long enough to get over that refractory period and be ready for them. But I don't know that. I'm just guessing. Yeah.
So there's a fundamental difference between aquatic smellers and terrestrial smellers. We have this mucus layer that intervenes between the odor and our receptors, which would add a whole extra set of physical constraints or filters, whatever I think. Do you think about that? I mean, you've got a lot of aquatic examples. Are you also involved in doing terrestrial?
Yes, but not vertebrates. So we've worked with moth antennae.
Well, those have they also directly interface with the environment. So
They do. They don't have. But what's interesting about insects is that in air, the diffusion coefficient of molecules is really high relative to the viscosity of the air, and the net result is that those odor filaments are really big in air compared to in water. And insects are really little compared to crustaceans, the bigger crustaceans we were looking at. And so it looks like the filament structure in air is big relative to the entire antenna on an insect. So they would be entirely in a filament or out of it. So what they see is sort of completely on or completely off. So it's a pulsatile. And other people like Neil Vickers have seen the same sort of thing, looking at insects and air odor plumes.
If you're a member or something with a mucus-filled nose, again, the large size of that plume filament is going to help you because the diffusion time doesn't matter as much. Right?
Well, the diffusion, yeah, it'd be interesting. I have never looked at invertebrate things where you have to have, you have rapid diffusion in air and then slower diffusion through the aquatic mucus before it gets to the receptor. So that would be interesting. You may know much more about that and can fill him in. I will mention one thing about the moth antennae before I shut up about them, which is that we also looked at the flow through, between the sensilla on moth antennae, and it turns out with every flap of the wings, they have to flap the wings apart to draw the air through the antennae. And so each flap of the wings is another pulse of air through the antennae. So they're getting a pulsatile sample as well.
And one more question. So, in vertebrate noses, we actually create a different Reynolds constraint when we sniff. I mean, our noses are enclosed and we're operating actually in sort of a different fluid environment than the rest of us. And I was wondering if you had any thoughts about that. Had you ever thought about that, or do you have any? You did allude to this in your talk that most olfaction is done without any olfactory physiology experiments are done without any thought to how odors are presented to olfactory receptor neurons into any of these problems. And I guess my question is, what do you think, or have you thought about what effect that might have on our understanding of olfaction right at this moment?
Yeah, I think all the studies about the kinetics of olfactory neurons, if you don't know exactly the kinetics of the arrival of the molecules, then I don't know what you make out of the kinetics of the neural response to it. And at least for the sort of crustacean literature I've looked at, people, and a lot of the insect stuff, they seem to just have sort of these streams moving past the antenna at some velocity, and then they flip a switch so that the stinky water comes in. And so it's really nothing like what the real antennule's doing. So I would love to see what the neural responses are like if you were to present odors the way they actually encounter them. So yeah, I don't know. I mean, what are your thoughts about that?
Well, I don't know. No, but I guess my sort of overriding question also is, why do you think we've evolved in air these noses that can only deal, like we don't have these lung appendages for dealing with odors, and yet we might be able to sample more of a plume if we had it?
Yeah, I mean, I don't know. Maybe it's very. I mean, once you're in air, I guess the filaments are big relative to your nose sensor just because of the physical medium. And if it's important, and I should ask you guys who study olfaction or neurobiology, why it's important to have a discrete sample someplace and then a discrete sample someplace else, rather than just sort of continuously monitoring. But I would think, taking a pulse of air into your nose is just as much a sniff as doing that.
Yeah, but you sample considerably less of the, or if you get a different sample with plume structure.
Yeah, and I guess you're sort of, if the air is moving slowly enough and you're breathing quickly enough, you're creating your own flow field too, which is a little different. So I don't know if just all this sniffing is important relative to the refractory time of neurons so that they're ready to deal with a different odor signal than the last one, or whether it's also important just that you know what's over here and now you've walked over here and you can compare. So, yeah. Yeah. Thank you.